Hello, welcome everyone. My name is Mohamed Koronfel, and in this talk I am hoping to give you an overview of the techniques at Beamline B24, an overview also of our workflow protocols, and some of the updates that have happened at the Beamline over the past year. Before I begin, also, it's important to note that this is one of two-part talk. The second part will be given by my colleague Ashna. We'll be discussing sample preparation protocols at our Beamline. In this talk, I will focus more on the techniques, and namely, we are a correlative light and X-ray microscopy Beamline. We have Superresolution microscope, Superresolution fluorescence microscope, and we have an X-ray microscope. And we do correlative imaging of biological samples in cryogenic conditions using these two techniques. Just as an introduction, our Beamline B24 is part of the biological cryoimaging group, one of the 8 groups that make up Diamond Light Source. Together we are with EBIC in this group, and you can find more details about our Beamline and the techniques that I'll be talking about on the website, on the Diamond website, diamond.ac.uk/instruments and then /biological cryoimaging B24. This is the core Beamline team. This was the core Beamline team about a year ago, and over the past year or the year and a half, our team has grown considerably, and you can find also more details about the Beamline staff also on the website. Over the past year, during the pandemic lockdown, we have been operational at B24. We have been involved in COVID-19 research, and we've worked to address some of the bottlenecks in our workflow to make our data collection easier and more user-friendly, and we have disseminated and published our protocols throughout this workflow, and I will give more details about these protocols later in the talk. So if we have a look at imaging life, observing life, and we have a look at a spectrum or looking down at an organism all the way to proteins and individual atoms and what techniques are available to use, what we do at Beamline 24 will fall around this region here, where we have a fluorescence microscope with a resolution of about 250 nanometers, and we have an X-ray microscope with a resolution of about 25 nanometers. Now, this resolution is not as high as what can be achieved with an electron microscope of about 5 nanometers, but there are plenty of advantages for using X-rays over electron tomography or electron microscopy, which I will be discussing also in the talk later. So, as an overview, this is our protocol. It has divided in 3 main sections: sample preparation, and that's what will be covered by my colleague Ashna in the other part of this talk. In this one, I will be covering the data collection on our fluorescence microscope, which is called SIM, or structured elimination microscopy, and our soft X-ray tomography microscope. And also I will be briefly discussing some of the data processing steps like correlation, and some data analysis and data deposition at the end. So these are images of our 2 microscopes, our 2 techniques. On the left-hand side here, this is our structured elimination microscope, or SIM for short, and it enables us to do 3D imaging of fluorescently labeled species. This could be pathogens, organelles, particles, etc. Inside cells under cryogenic conditions. And we have our X-ray microscope, which is essentially our beamline, where we're using the synchrotron as the X-ray source, and we can do 3D imaging of cellular ultrastructure using this microscope of the same regions of interest that were collected on the SEM, and we can do correlation between the two data sets. So to begin with, just a little bit more details about our super resolution fluorescence microscope. We have a CryoSEM. It's a bespoke microscope that was built here to enable imaging of cryopreserved structures at high resolutions, well beyond the conventional fluorescence. And we image vitrified samples, so in cryogenic conditions, and we have excitation beams in the visible spectrum. We can image samples that are around 10 to 12 micrometers in thickness or more on the SEM, but to match the X-ray microscope, this is around the thickness that we can image with the X-rays. And we have a resolution of 200 nanometers using this microscope, and this is limited by the numerical aperture of the objective lens that we're using. In the future, we are looking also to develop another super resolution fluorescence technique called D-Storm, and we'll be looking to also develop that for cryogenic samples, and that will enable us to reach resolutions of tens of nanometers. And these are just examples of conventional fluorescence using a wide field elimination compared to what we get with our structured elimination microscope. You can find more details about this microscope and all the technical details in this paper reference at the bottom here. Now moving on to our second microscope, the X-ray microscope. This is a schematic of an overview of our beamline. So we have a bending magnet, which is the source of our X-rays. So this is a bending magnet at the electron ring, and electrons, as they pass through the bending magnet, they lose energy in the form of X-rays, which then passes through the beamline components, which are mainly beamline slits for beam profile, a focusing mirror, and then we have our monochromator, which enables us to select the X-ray energy of interest, and further slits here for the beam profile, and then we have our actual microscope. So this area here that's enclosed in this gray rectangle is the actual X-ray microscope, and it's a chamber that's maintained under high vacuum and then under cryogenic temperatures. And it has a condenser, capillary glass condenser lens that focuses the beam on our sample. And then we have a zone plate objective lens that then focuses the image onto the detector. And if we look through this chamber here, this is a top view, and in the purple here, this is our capillary condenser. This is the glass tube, and this is the side view. You can see it also here. And on yellow arrow, this is our sample. And the blue arrow, the dark blue arrow, is our objective lens or the zone plate. And again, this is the side view. So this is our zone plate. Our sample is sitting inside this shield here, which is a cryo shield, helping maintain the sample stay at cryogenic temperature. And we also have a sample storage area where we can store a total of 4 samples inside this cryogenic chamber. So we can load 4 samples into this chamber at once, and then we can switch between those samples more easily without the need of any intervention. It's also worth it to note that inside this chamber we have also an optical microscope. So this is an optical objective that you see here, and this enables us also to get an optical microscopy map of the sample, which enables us to identify areas of interest and regions of interest easier before going into imaging with X-rays. So a brief of the techniques, or sorry, the specifications of this technique. The X-ray microscopy, we can image cells in their near native state. So cells are cryogenically preserved There is no need for any chemical fixation like in EM, in electron microscopy, or any staining or sectioning. There's no need for sectioning cells as well. We can image through full cells. So these are the main advantages over electron microscopy. We also have on the Beamline dual access tomography, and we have developed automated data collections of tomograms or tilt series. So we can collect X-ray images or projections at different angles of our sample and reconstruct this stack or series of images into a 3D volume. And we can do these collections automatically and do a reconstruction automatically through a pipeline, data processing pipeline that we've developed here at the Beamline. We use an excitation beam or an X-ray beam of 500 eV. This is above the carbon edge, the carbon K edge, absorption edge of X-rays, and below the oxygen absorption edge. So what that means is this is also called the water window. And what that means is we get a preferential absorption from carbon in our samples. And so carbon structures like the cellular structures and organelles, carbon-rich structures will appear darker on our image, while the cytoplasm or the surrounding more water regions will appear lighter in color. And so this is what generates the contrast in our images, which is called the absorption contrast. We can do tilts up to 140 degrees. We can image through samples of 10 to 12 micrometers in thickness. And we have two objective lenses that enable us to image with a resolution of either 25 nanometers or 40 nanometers. In the future, we're currently also developing phase contrast imaging, which will enable us to get better contrast from samples with less absorption contrast. We have the X-ray optics ready for the microscope for this, and we're developing a design for a new monochromator grating that will enable us to move to high energy of 2500 eV, where we will be doing this for phase contrast imaging. Also, on the future developments, we are also looking as part of the Diamond 2 project to move into a biosafety level 3. We are currently hosting samples of category 1 and 2 biosafety levels. We can prepare them at the Beamline and we can use them, image them with our techniques. In the future, we're looking to also be able to image level 3 samples. So this is an example data set of some of the data that is possible to collect with our X-ray microscope. And what you're looking at here is an X-ray mosaic. So that's a mosaic of 5 by 5 images. Individual image is about 10 micrometers by 10 micrometers. So this whole region is about 50 by 50 micrometers. And then we collect tilt series from one field of view. So this is about the 10 by 10 micrometer field of view that we have with our X-ray microscope. And we can collect tilt series, for example, as you can see with this figure here. So this is 120 degrees tilt series at steps of 0.2 degrees. Total data collection time is about 20 minutes for this. And this tilt series would then reconstructed into a 3D volume using reconstruction software iMod. And this is also a video of that reconstruction of looking at the 3D volume. So these are images through the depth of the cells. So as you can see, if I pose this, some of the cellular ultrastructure here, so the lipid droplet here, this is the nuclear membrane and the nucleus on this side. The cytoplasm is on the left side with some features here visible like the mitochondria, endosomes, and endoplasmic reticulum. So before moving on into the data processing, I just wanted to give an overview of certain criteria that might influence our data collection strategy at the X-ray microscope. So this is an image from a review paper here, which is a general tomography or hard X-ray tomography actually paper. But so the main things that I wanted to highlight is that the image settings, so we can adjust the exposure time depending on samples, the number of projections, etc. And so that can impact our temporal resolution. So what you see here is the certain criteria like the contrast. As I said, we already do that by imaging at the water window, choosing a beam energy at the water window. And our sample material is biological samples. So throughout these criteria, these are the different parameters that can impact these criteria. So for example, the spatial resolution, X-ray optics, we have also a choice of two optics, a 25 nanometer zone plate or a 40 nanometer zone plate, for example. So now moving on from the data collection into the data processing, and the first step here is the correlation. And just a quick overview, basically collecting the data on our two microscopes. When collecting the data on our two microscopes, the sample might not be in the same orientation when you move it from one microscope to the other. There's also differences in the resolution. And so to do the correlation, we have an in silico step, which is done to overlay the two data sets together. So as an example here, this is an X-ray image, and these are fluorescence microscopy images. And then by overlaying them together, we get the merged sequence here. And by doing this step in 3D, as you can see here on the right-hand side, we can see looking through images through the volume or through the depth of the sample. We can look at the X-ray data correlated with the fluorescence data. And we can image up to, with the fluorescence microscope, we can image two channels as well. So we can correlate two different fluorescence channels with our cellular ultrastructure data from the X-rays. And just a brief overview of the process. Again, you can find more details about it in this paper here, which describe the software that we use, ECCLIM. And generally, the process involves, first 2-day overlay of images. We start with X-ray mosaic image, and we overlay it with the bright field optical image. And then from then we can overlay the fluorescence data, and then we can then correlate that to the actual X-ray tomogram. Now moving on, just briefly on the analysis. This is just one example of a quantitative correlation analysis, which was looking at the sizes of different exosomes, and from the X-ray microscopy data, and correlating that to the intensity of the fluorescence. So this is one of the study that we had of the Beamline looking at vaccine candidates. We usually use Fiji, iMod, IMERA, and Servos, which is a Diamond software for segmentation of data sets, and we'll have more details on this in an upcoming publication. Finally, the last step of our protocol is the data deposition, and so we expect all data collected at our Beamline to be deposited in online archives. And this is to enable access to the data to the scientific community, by the scientific community, unhindered access, and lead to independent validations, and allow the use of this data in comparative studies or educational activities, or to progress research. And so we have have a protocol with in collaboration with EBI, the European Bioinformatics Institute, describing data deposition steps into their online archives for this type of data. So again, back to our workflow and just an overview of the protocols that are published and available currently, describing every step of our sample preparation and data collection workflow here at B24. So as I mentioned, the blue section here, the sample preparation, will be covered in another talk, and it's also covered in this publication here, the steps from cell culture, vitrification, and assessment of samples. The data collection on the SIM, the fluorescence microscope, and the X-ray microscope are described in this paper here. We have a Jove paper here describing the steps for collecting data in SIM. The Optica paper here describes the technical specifications of the microscope. Another Jove paper describes the data collections on the X-ray microscope. And we also have the GitHub releases by our software scientist, data analysis scientist, Tom Fish, on GitHub, of different software that he developed for automated data collection and reconstruction. Then for the correlation of the data between the two microscopes, we have a STAR protocols paper describing the steps for this part. And we have the protocol describing data deposition of the data on the online archives of EBI. And as examples of some of the data sets that have been collected at our Beamline from different biological questions or different applications, we have those 3 papers here that cover different areas of biology or science. Finally, before I finish, this is just an overview of how to gain access to our Beamline. The first step is to contact one of our team members, which you can find us on the Diamond website. And then the second step is to submit a proposal. So after discussing with one of our team members, we'll be able to help you and guide you through the submission procedure. And there are different proposal submission routes at Diamond. These are the different 4 routes here. And at B24, we have access to the standard access route for our X-ray microscope and the rapid access for our SIM microscope, for our fluorescence microscope. And it's important also to, if you would like to use both microscopes, to submit the 2 types of proposal. And it's important for us to have discussed this beforehand to make sure you're allocated time on both microscopes for the same samples. Finally, I would like to thank all the staff at Diamond Light Source, and special thanks to Liz Duke, Jeff Irwin, and Eva Pereiro, and also to Mick Phillips and Ian Dobbie from Micron Oxford, who developed our SIM microscope. Thank you very much, and you can follow more details and updates about our Beamline and our research on Twitter at the B24 Light handle. Thank you.